Well, hello, it's Bruce Williams again, and today I want to present the second lecture in my series on ultrastructural pathology, in which we're going to talk about cellular degenerative changes. A few things to remember about cellular degeneration. Number one, you're not going to diagnose necrosis on EM because when tissue is necrotic and devitalized, the osmium just globs onto it and it looks like a big black blob. So there's nothing to diagnose in the realm of necrosis. You will often see normal tissue on EM, and then we have degenerative changes which make up the abundance of pathologic pictures which we see in electron microscopy in animals. Now, we're very lucky in that cellular degenerative changes are very stereotypical, and it doesn't matter whether you're looking at an enterocyte or a neuron or a lymphocyte or any cell in the body. They basically go through the same morphologic change at the ultrastructural level, which allow us to recognize that this cell is sick. So we're going to start with my favorite, the prototypical epithelial cell, which we spent a lot of time with in the first lecture. Let's just quickly review all the parts that we can see and where we can see them. Remember, epithelial cells have a very directional status to them. They may be a secretory or absorptive or whatever, but everything is generally put in the appropriate place, and the whole cell is locked down in between other similar cells and onto a basement membrane. Okay, let's start at the luminal surface. If this was an enterocyte, this particular cell has components of a number of epithelial cells, so it's an imaginary cell. Okay, first thing we see is a cilium right here. Remember, all cells of the body have at least one cilium, so I'm told, but epithelial cells in certain parts of the body have lots of them. But this one only has one lonely cilium. Here at the bottom is the basal body, which anchors it to the cell. The apical surface of the cell is covered with microvilli. All epithelial cells have microvilli. We have tight junctions to make sure stuff does not go down the outside of the cells. And we have a belt desmosome. And here are the little keratin filaments projecting into the desmosome. Okay. This cell has a lot of mitochondria. This particular one is oriented at the bottom near the basement membrane, which is what we characteristically will see with the proximal tubular epithelium, something that has a profound secretory activity. So, but we just put them all down here so they don't clutter up. We have a nucleus with a nucleolus with some heterochromatin, with some euchromatin, and then we have smooth endoplasmic reticulum here and in here. We have a Golgi apparatus. We have what looks like to be maybe a multivesicular body, okay, which are essentially phagosomes with effete bits of nucleus. They'll float around. Here's the rough endoplasmic reticulum with those ribosomes that stud it. Remember that rough endoplasmic reticulum produces protein for export from the cells, and the free ribosomes and the cytosol will produce the protein that's used within the cells. This nucleus also has a nuclear envelope. Remember, that connects to the smooth endoplasmic reticulum. So, this is our prototypical cell. And what we're going to do, we're going to be mean to this cell, and we are going to deprive it of oxygen for a couple of milliseconds. It doesn't take long. Okay, oxygen is required to produce ATP, which is required to run all of the processes of the cell. Okay, so when we shock this cell by withholding oxygen, make it hypoxic for just a tiny little bit of time, we will start to see changes in the cell which are associated initially with reversible and then later on with irreversible change. Okay, so the first thing that you will notice, and really there isn't a whole lot of of change here, but this cell has been stressed. And the first thing that will happen is that the sodium-potassium pumps on the cell membrane and within certain organelles will stop functioning because they don't have any energy to power them. And so sodium will stop being pumped out of the cell, it will accumulate within the cell, and water will follow. And the first place that it accumulates, before it accumulates in the cytoplasm proper, is within the lumina of the smooth endoplasmic reticulum. So this is often the first thing that you will see that says that the cell is stressed. And remember, look closely. Remember I said that one of the very characteristic signs of cells, the early parts of cell swelling, is that the nuclear envelope becomes dilated. And that's what we're seeing here. You know, I've seen a lot of smooth endoplasmic reticulum. I couldn't tell whether it was dilated or not, but when I saw the nuclear envelope being dilated, I knew that I had a stressed cell. But the organelles are all in the right place. They're not going anywhere. The mitochondria are here at the base in this cell. The surface substructures of the membranes look pretty good. Cilium looks good. The microvilli. All we have is this little dilation here. The cell might swell a little bit, bulging into its neighbors if it wasn't locked so hard in place. But we will see that a little later on. Now, if we looked at this, you know, I'm going to go back and forth so you can see the change. Here's normal. Here's abnormal. There's a little bit of condensation of the chromatin, which probably results from some of the heterochromatin breaking loose and coming into the center of the nucleus, but it's a minimal change. It's almost imperceptible on EM. I don't know if I would be able to see that because you don't have the ability to go back and forth from normal to abnormal. But that's all that's going on here. And if we look at that on transmission electron micrography, in this section of a liver cell, here's a nice nucleus here. The nuclei are still intact. It looks pretty good. Okay, we see multiple fat globules. Remember, you don't have a lot of cells that will have fat in them, and so liver is one of them. We have a lot of mitochondria. We can't get too close because we can't see the cristae very well in this. We're a little far away. But what you can see is that all of the smooth endoplasmic reticulum is dilated because it's accumulating water. And if you're not sure, if you're saying, well, maybe that's just, because this tissue is dug out of paraffin, then look at the nuclear membrane. Okay, to me that says, hey, this dilation is real. This cell, obviously we can right the ship. This is not irreversible change. We give this cell oxygen or whatever it needs to start making ATP again. It's going to pump that water out, okay? But that's the earliest and the most perceptible early change. The nuclear line, I said, don't make too much out of it. In early change, you will see that the nucleoli tend to marginate. The fibrillar centers tend to disintegrate, and then the granular portion of the nucleoli. And I want you to take a look at the cytoplasm. Looks darker than normal, doesn't it? And that's because those swelling smooth endoplasmic reticulum are compressing them. Now, when we put more water in, it's going to become very lucent again. But right now, it's compressing all of the fibrils and the glycogen, whatever's in there, sort of together, and gives the appearance it's darker, but it's really just sort of compressed because of the dilation of the smooth endoplasmic reticulum. Okay, this particular case was a case of carbon tetrachloride intoxication. Okay, so we're going to be a little meaner to this cell. Sorry, cell, but it's important stuff. And now you can really start to see that there are a number of changes going on here. First thing that you'll notice is the smooth endoplasmic reticulum is still dilated. We will probably have more lucency of the cytosol as all that stuff that was compressed starts. The cell is starting to swell. Probably it should have been drawn as being sort of bloated on the sides, and you can often see cells at this point, they are, even though they're epithelial cells and they're bolted together, they're swelling and they're sort of compressing the ones next to them if they're not similarly affected. Another major change that you're seeing here in the late stages of regenerative change is the loss of the surface substructure. That cilium, boop, it's gone. Okay, I'm not sure the basal body is probably floating around somewhere, lost and depressed because the cilium's gone, but it's just gone. And remember all those microvilli? Well, they're gone too. The actin filaments that form the structure have probably changed from a fibrillar to back to a globular, and so when that happens, they just sort of go away because they're just linear elongations of the cell membrane. They disappear, so the cell membrane becomes somewhat undulant here. There's a loss of polarity of this cells. All of these organelles, all these mitochondria that were nicely oriented at the bottom of this cell are now starting to float free because the cytoskeleton is breaking down. We've lost the actin, so the cell's starting to lose its traditional shape. The tubulin components of microtubules have disassociated, so the microtubules have broken down, and the cells that were attached and moving along them or whatever are just now sort of free floating. So you're starting to see maybe the Golgi will float away from the nucleus, and the mitochondria will float away from the part that they need to congregate because it's a highly metabolic active. If the nuclei is normally basal, like we'll see in secretory cells, it may be floating up at the apical surface and intermix with whatever granules that normally. So the late stages of reversible cellular degeneration are characterized by disassociation of the cytoskeleton and changes with that. Now, the other thing that is extremely prominent right now is that there are profound changes in the mitochondria. At this point, we call this low amplitude swelling. It's a reversible change in the mitochondria, but what we see is there is swelling within the lipid bilayer and within the cristae. So the cristae, which are normally long and thin, are becoming sort of fatter, and this causes a increase in density of that matrix, which is getting compressed. So it's actually darker than normal. So this is more cristae swelling than a true degeneration of the mitochondria. And I think that we've covered all the changes. That the desmosomes are still intact, the cells attached to the basement membrane. You may start to see an accumulation of secondary lysosomes, increase in multivesicular bodies as some of the membrane is starting to show wear and tear. And when we look at this, at least we look at the mitochondria and the cytosol on transmission EM. Here's that low amplitude swelling. Okay, yes, it's characterized by a condensation of the matrix, a swelling of the cristae. You can see that the smooth endoplasmic reticulum is significantly dilated here, and opposed to the previous slide where the cytosol was condensed, it becomes light and lacy because of all of the water that has been absorbed by the cell. Now, this is pretty much the end of the reversible change. We're about to go into some irreversible changes where that cell is really not going to be able to come back due to the damage. And so as we move forward, we start to see a couple of other changes. I think the first thing we'll see is you will see the nucleus. You will have dissolution of chromatin. The nucleoli are totally gone. Both fibrillar and granular parts have broken down. This cell is probably struggling to maintain any form of homeostasis, whether it's sodium, potassium, or it's calcium. And the changes in the mitochondria, which are now free floating around the cell, are changing from this low amplitude swelling to a diffuse swelling of the entire mitochondria. If this was one with low amplitude, look how big these have gotten. So you have a lot of fluid that's accumulating within. The cristae are breaking down, and you see an accumulation of these dark deposits of calcium. Calcium is a poison for mitochondria, and you get too much, and then they can't do their job. The other thing that you're starting to see, we don't see it on the outside, but around the organelles, you're starting to see a breakdown of that lipid bilayer. And remember that the lipid bilayer, and we'll look at this in some other pictures later on in this lecture series, but that lipid bilayer is very strictly regulated, with the hydrophilic parts on the inside, like the bologna on a sandwich, and the hydrophobic parts are on the inside, the hydrophilic parts are on the outside, okay? And it works very well. But if you break that down, then you get the hydrophilic and hydrophobic, which have an attraction to each other. One flips over, they become very sticky, and they start to wrap around the outside of each affected organelle. So all of this probably was part of the membrane of this degenerating mitochondria. And that's part of either high amplitude swelling, or you can see that in cells that have been poisoned by a number of drugs which have a detergent effect on the fat in the cell membrane, or any type of peroxidation by agents which cause damage to unsaturated fatty acids, cationic detergents. So anything that damages the organelle membrane or the high amplitude swelling will cause the formation of these lamellae, which some people call zebra bodies. Zebra body is a term that's used a lot in ultrastructure and is never really used appropriately, but I've heard them called zebra bodies. So they're just lamellae of effete and broken down membrane. If we look at this on transmission EM, the entire mitochondria is big. It's swollen. There are only a few cristae left, and we start to see these accumulations of calcium within the matrix. We still have this very lucent cytosol. There's these many dilated profiles of smooth endoplasmic reticulum. I'm not sure what the V is for. I sure hope it's not for vacuole. Okay, and then finally, we're at the end stage of the cell. We have irreversible change. We have the high amplitude swelling, the accumulation of the calcium within the matrix of these dead mitochondria. There's rupture of the nuclear membrane and an influx of fluid into the nucleus. We have total lysis of the chromatin. If you start to look around the outside cell membrane, there are multiple ruptures within the cell membrane which let in even more fluid. The only thing it's still hanging on to the basement membrane, and one of these desmosomes is gone. The other one's still there. Desmosomes are really tough. They're usually the last thing to hang on, at least in an epithelial cell, in stages of, but then eventually they will, the cells will swell so much they'll rip apart. The cell will release from the basement membrane and will float off and just become part of the necrotic debris. Cells that release from each epithelial cells really are social creatures. They need to be connected to each other. They need to be connected to a basement membrane. If they release from that, even if they're perfectly healthy, they will undergo degeneration. So fluids pouring in, it's all done here. There's no coming back from this. So those are the stages of traditional degeneration. Notice I haven't said necrosis, because if this was necrotic, it'd be all black and blobby and we wouldn't be able to see anything. So those are the stages of cellular degeneration that you should be able to identify. You should be able to put in a description to justify a morphologic diagnosis of cellular degeneration. As I've said before, and always bears repeating, it doesn't matter whether this is an epithelial cell or whether it's a fibroblast or whether it is a macrophage, because all cells break down the same way. The organelles go through the same degenerative changes, and it really doesn't matter. So it's a pretty easy concept. There are only a few steps. They're very stereotypical. So I'm very confident that even with this brief instruction, you will be able to recognize degenerative cells. And as we go through these lectures, we're going to look at a lot of cells. We're going to be able to identify what cell type they are, are they degenerate or not, before we start adding in the other fun things like viruses and bacteria. And hopefully I will remember to point out what makes a cell, a particular cell. Where is it degenerative? How do I know it's degenerative? And so on further down the road. Now, there are, in the last number of years, there have been a number of changes to the nomenclature, and we have identified a number of years ago a second way, and they've been subdivided, but I like to keep it fairly simple. And you have necrosis, which is usually a pathologic change due to injury of any number of types. Okay, the other thing that you see which causes cells to degenerate is a normal function, which is called apoptosis. My pet peeve, the second P is silent. It's not apoptosis, although that's a great sound and it sort of makes you think about cells exploding, but it's really apoptosis. And we see this. There's a lot of cells in the body that undergo periodic cell death and replacement. It's, as we said before, it's called apoptosis. For example, lymph nodes have a high rate of apoptosis. Your body is constantly shifting in its ways that it combats, foreign antigens, recognizes antigens, combats foreign agents. And obviously the antigens you're exposed to in the early part of your life, you don't really are exposed to later on in your life. So your body's going to shift, and you're going to, those cells that recognized, for example, pine pollen when you were a kid like me growing up in Georgia, I don't see that anymore. So all of those cells, we don't need them. And the body will normally turn over a lot of cells. A lot of you have apoptosis in the intestine as the enterocytes mature. You have apoptosis that women go through on a monthly basis, where they form a lining of the uterus and then they slough it again. So this is a constant process as the body renews itself. And the morphologic changes are very different than what we see. They don't go through this type of injury and death. It's a programmed death, which looks very different. Okay, instead of the cell losing its cellular integrity and water coming in and it exploding, for lack of a better word, the cell starts to break down and it breaks into small pieces. Okay, here is a prototypical cell. It's probably, this one's round, so maybe we'll call this a lymphocyte, but it's got a whole lot of organelles which lymphocytes don't have, but that's another lecture. But what will happen is you have a couple of choices. You can go through traditional necrosis, and all the changes we have with the mitochondrial swelling, the ruptures in the nuclear membrane, the lysis of the chromatin, and then, as we've seen before, we have breaks in the nuclear membrane. That's more of a traditional necrotic change. We're going to undergo apoptosis. And what I want you to see here is one of the changes that you see is that you have these crescents, which are formed from breakage of the linkages of the DNA. So the DNA will break down to anywhere as little as 120 base pairs, and it sort of orients itself around the outside of the inside of the nuclear membrane. Okay, and then at the same time, the cell will start to break up into little bits, and the bits may contain some organelles. It may contain a fragment of this nucleus, but at no time does the nuclear membrane rupture. You don't have the influx. The organelles really look very good. They look like they're in mint condition, except the nuclei are a little ragged with these crescents. And then it breaks up into little pieces, and the little pieces are generally phagocytized by macrophages or other phagocytic cells, where they are broken down, and then you will end up with a secondary lysosome with some lipofuscin in it from breakdown of the membranes of the various organelles. Okay, if we're in the lymph node, then we would call these lymphoglandular bodies, and the macrophages, which are resident in the lymph node, that ingest these, would be called tingible body macrophages. And the little basophilic dots in their cytoplasm, which is why we recognize them as tingible body macrophages, are actually little bits of effete apoptotic cells. Okay, I don't lecture on general pathology. There are a vast number of triggers for apoptosis, including the caspase cascade, including fast ligand activation, and many more. And I will leave that to the people who teach on general pathology to explain that. But it is a very characteristic, even on ultrastructure, this is very characteristic. We're looking at a case of leukemia in a mouse, and whenever you have tremendous proliferation of lymphocytes, you pretty much always have tremendous death or apoptosis of lymphocytes as well. Any really hot neoplasm, there's a lot of turnover, and a lot of that's due to apoptosis. So what you can see here is these are cells here. It looks like a vessel with an endothelial cell nucleus, 2 erythrocytes, and we'll talk later on about how erythrocytes are your friend. But we have, maybe this is a lymphocyte which is just starting to break down. You can see how the chromatin has started to form these crescents on the inside of the nuclear membrane. And here is one which is going through the exact same process that we looked at in this diagram. It is starting to fragment into pieces. Here you can see little pieces, but if you look closely, you can see that all of the organelles, at least the ones that you can make out, this mitochondria, this rough endoplasmic reticulum, this, I don't know. That's one of those things that you got to be careful. This and this, those could be primary lysosomes, but remember, try not to overinterpret. This little fragment of a nucleus, it all looks really good. Okay, and then there's going to be a macrophage that comes in. I don't see one here, but all these little bits are going to be gobbled up and broken down. And that's what apoptosis looks like. In the case of human Burkitt's lymphoma, once again, we have neoplastic B cells with a high rate of turnover. Here we have one that the nucleus probably looks pretty good, or over here. Then we have one that's undergoing apoptosis. You can see the formation of the crescents. The nucleus is breaking up into fragments. Little blebs are blebbing off of the cell, but the organelles look great. And then up here, you probably have, looks like this may be a phagocytic cell, like a macrophage. It has engulfed one of these little blips of a degenerating cell, and it's starting to break it down. Notice how everything's starting to get blacker and blacker and blacker, and that's what happens until it's just a big blob of lipofuscin or spheroid. Okay, great preservation in a cell that's breaking up. We're dealing with apoptosis. And just a better picture of macrophages, which are digesting apoptotic bodies of cells in a prostate carcinoma from a rat. Okay, here is your macrophage. And the macrophage, here's the nucleus. Macrophage has ingested 2 fragments of the degenerating prostatic epithelium. This one still looks pretty good. This one has a big crescent. You can only see a little bit of the residual euchromatin, but there's a nice nuclear membrane here. There's a lot of endoplasmic reticulum. And this one has been partially digested. Everything's starting to condense. Everything's starting to lose its integrity and turn black, and it really sucks up the osmium on this. So it probably ate this one a little bit before it ate this one. So that's apoptosis. And that brings us to the end of this particular lecture on cellular degeneration. I hope you enjoyed it. I hope you will hang on for a couple more lectures. In our next lecture, we'll probably do 2 lectures. I'm going to split it in half on the various clues that tell you what type of organ you're in when you do an ultrastructure. How do you tell a gastric epithelial cell from a pneumocyte or a kidney cell or something like that? So I look forward to putting those on the video shortly, and I hope that you'll come back and watch. Until then, I wish you good health. I wish you to stay indoors, stay safe, and we'll see you again.